one's here. All right. Couple of reminders. We have lab this week. It's outside, rain or shine. Should be shine all week. Today, Monday, lab looks out again. Should be very pleasant out there. Tuesday and Wednesday, the temperature gets back up. Then Thursday has that cold front coming through, so we'll see if that happens. Ready? We'll see if that happens. But we're outside, we're on campus. We'll meet in the lab, we'll take a quiz, we'll go over what we're gonna do, and then we'll get out and measure it. This is one where when you finish, you're done, we're done for the lab. You are gonna work in pairs, and the lab handout has a data sheet. That data sheet will get turned in. So just make sure at least one person in your group has that data sheet that you can record the data and give it to either Dr. Skipper or myself or Cody. All right? So that's for lab. Friday is our exam number 2. That exam will go live sometime in the morning on Friday. Sometime in the morning, meaning like 5 in the morning. You have all day to get it done. You have to have started it, I believe, by 11:30 in the evening. So it's going to be a time limit. So once you start, the timer starts ticking. I don't think we're going to do a word list. I think the definitions will be more multiple choice. So I'll give you the definition, and there's going to be 4 words that you can choose from. All right. If you have any questions, please email me. So this is where we left off. So we finished up the frequency-dependent selection. We kind of got through it a little bit quickly, but because of that, I just kind of want to go over it again. So frequency-dependent selection is a mechanism that keeps variation in our populations. So it's a way in which that promotes genetic variation by making sure that we have more than one phenotype. So it doesn't just have to be one phenotype. It could be multiple phenotypes, I should say. It doesn't have to be 2 phenotypes. It could be 3 or 4 or 5. All right? So with our phenotypes, we're always talking about, a discrete type of phenotype. So long arms versus short arms, for example. But you have to realize that the arm length is actually more of a quantitative trait, and thus you can have different fitnesses based on different sizes. With this frequency-dependent selection, it's saying that our fitness changes based on the frequency in the population itself. So it's not necessarily how well your adaptation does in the environment. It's more about how well does your adaptation do compared to everything else in the population. So, if you're super common, oftentimes you're going to be less fit than if you're super rare. All right? And because of that, if you're rare, your fitness is going to be higher than if you're common. And you get an increase in frequencies, and thus, as the frequencies increase, you become more and more common, which drives your fitness down. And it gets to the point where we have this balance, where fitness of the 2 phenotypes are basically equal, and we stay there. All right? If our trait increases in frequency, selection pushes us back down. If our trait decreases in frequency, selection pushes us back up. That it gives us a way to maintain more than one phenotype in our population. All right? So any questions on frequency-dependent selection? It's out there. It's out there. All right, so a quick summary of this stuff. So this is the sources and causes of variation. So according to Fisher's fundamental theorem, adaptive evolution depends on the genetic variation on which our selection can act. All right, so we have to have that heritable variation. That goes back to Darwin's tenets. We have to have that heritable variation. That heritable variation actually has to code for the phenotypes that selection is favoring or that selection is favoring. All right? So how do we get that variation? Well, mutation. That's going to be our ultimate source of variation. That's what creates it. And then selection and drift in gene flow can increase, decrease its frequency. We also have several other processes that cause an increase in genetic variation, including sexual reproduction, disruptive selection, and the most recent one that we talked about, frequency-dependent selection. All of these increase our genetic variation so that hopefully, if the environment changes, our organism has some heritable variation that allows it to survive. But we can't talk about processes that increase variation without talking about those that decrease variation. And for that, we have inbreeding. So breeding among closely related individuals. That causes a decrease in genetic variation by exposing our homozygous individuals, especially the homozygous recessives. Genetic drift decreases genetic variation. If given enough time, alleles will be fixed in our population. And also stability, stabilizing and directional selection. Those are the other two. All right. So go back. We talked about some of these in previous lectures. If you want more information, you can go back to those. Also in this lecture, since we're talking about genetic variation, we have the possibility that selection creates ecotypes, which are genetically distinct populations or genetically distinct subpopulations. A lot of times, these are the same thing as subspecies or races, all right, where those subspecies and races have phenotypic differences, but a lot of times they don't have to be. They can look just like any other individual in our species. It just so happens that those individuals in a group, wherever they live, are genetically distinct. What favors their formation? Couple things. Strong selection pressures are number one. Those individuals must live in an area where there's a strong selection pressure. It allows evolution to push it down its own evolutionary path. Barriers to gene flow prevent they prevent mixing of our genetic code and eliminates that path that we're taking on. It just kind of mixes it all back together and eliminates it. So if we have barriers to gene flow, it allows evolution to go down its own path. Then the third factor that favors that is homogeneous and predictable conditions. So if our populations are in an environment that is pretty much constant, it allows evolution to take hold and push us down that path. If it's variable, if it's unpredictable, evolution's going to get mixed up. Evolution starts pushing us one way, the environment changes, and now it pushes us a completely different way. Sometimes, though, we don't have these conditions. We don't have homogeneous and predictable conditions. But our organism still has to be able to survive those changing conditions. So how do they do that? Well, they can do that with phenotypic plasticity. So they could have evolved the mechanism to alter their phenotypes when the environment changes. Now, the phenotypic plasticity is always going to be favored whenever our conditions are less predictable, when our conditions can freely change. Why is that? It's because you don't want to be stuck with a single phenotype. And if all of our variation, if all of our phenotypic variation is determined by the nature component, right? So by our genes itself, we're stuck. We can't change. So if the environment changes, that's it. We might die out. But if we have the ability to change our phenotype, then from one genotype, we can produce multiple phenotypes, and hopefully we have the ability to produce a phenotype that allows us to survive and do well in that new environment. All right, so if it's less predictable, and that is less predictable over time, it's also less predictable over space. So that is our fine-grained variation. We don't know where we're going to end up. We don't know where our offspring are going to end up, and it's a possibility that our offspring or us might end up in a completely different environment. All right, so you should probably know when we would favor genetic adaptation and when we would favor phenotypic plasticity. Kind of think about the different situations. Think about this variation, this environmental variation, and think about when one would be favored over the other. All right? Questions? All right, so that was the sources. Kind of gave you the background, lets you know what's out there. It was right after our environmental stuff. So now, how do we measure it? Because that's always kind of like the other thing. It's easy to kind of rationalize looking at individuals and saying, oh yeah, we have a range of sizes. That's the phenotype. But what if we don't have that? What if we can't do that? What if we're interested in knowing how much of our variation is determined by our genes and how much is determined by phenotypic plasticity? How do we go about doing that? Well, this is the topic of this presentation. All right? So, we're going to start with genetic variation versus phenotypic plasticity. How do we distinguish between the two? All right? Typically, we perform something called a common garden experiment. What this means is exactly what it says. We're going to take our individuals, they're from two different areas, and then we're going to raise them in a common garden under the exact same conditions, and we're going to see what their phenotype is. All right? So we could take them from two different environments, put them in a neutral, or we could do something called a reciprocal transfer study, where you take individuals from one habitat, put them into the opposite habitat, and vice versa. All right? So you take them there and you just grow them in the other's habitat, and you see the changes. All right? If our differences persist, then these are going to be genetic differences. If you see changes, and the differences no longer exist, or maybe the differences are less, then you know that that is due to phenotypic plasticity. All right? So, let's talk about this. I'm going to increase the size of this. All right. So our example, we will say, is a hill. And we've got trees or plants. All right. There we go. At the bottom of our hill, we get a tall phenotype. At the top of a hill, we get a short phenotype. And we're curious, how much of this phenotype, the height phenotype, is due to the genetics, how much of it is due to phenotypic plasticity? So what we could do is take them both and raise them in the same plot of land and see what happens. All right? We could do that. That's a common garden experiment. Or what we'll do here is a reciprocal transfer. Where we take the short phenotype, grow it down at the bottom, we take the tall phenotype, grow it at the top, and see what happens. All right? See what happens. So, this is what we do when we grow it. So we have bottom of the hill, and we have top of the hill. All right, so at the bottom of our hill, we have one phenotype that grows tall. All right. So the open boxes will be our tall, our hatched boxes will be the short. Will be our short phenotype. All right, so at the bottom of the hill, our tall plants, they grow tall. At the top of the hill, our short plants are short. All right, that's our normal condition, our natural condition. What happens when we grow them in the opposites environment? Well, let's say our tall plant, we grow it up at the top. What we might see is something like this. Our tall plant is still tall, at least taller than a short, but it's not as tall as if it was grown at the bottom of the hill. If we did our short plant and grew it at the bottom, we saw that we could see that our short plant then maybe grew taller. But again, not as tall as our tall plants. All right, so which part of this is due to the genetics, which is due to our phenotypic plasticity? Well, these differences that persisted, let's get that off to the side a little bit. Get that. So, these differences are our phenotypic plasticity. I'm sorry. Not those different. Those are genetics. They're the differences that persisted. We've grown them in the same environment. We still have differences in our height. That's due to our genetic. That's our nature component. The change, so this up here to this down here, that's our phenotypic plasticity. This to here, that's our phenotypic plasticity. So that change within a single phenotype, from short, we saw how it changed there, the tall, we saw how it changed. That's the amount that's due to our phenotypic plasticity. The differences that persisted is due to our genetics. Nature versus nurture. All right. Questions? Not too bad? We'll do that. All right. So I did have this. So one more slide, just kind of the example, seaboard. That's what we diagrammed up here. Hopefully you can kind of rationalize it. If you think about it, it kind of makes sense. Any difference that persists, if you grow them in the same types of environments, that's all going to be to our genetics. That's our difference. It can't be nurture, it can't be our phenotypic plasticity, because they're experiencing the exact same phenotype or the exact same environment. All right, so this is great when we have different phenotypes, all right, different phenotypes in the same species, and we might be interested in it. All right, I know one that was interested in snails, and that's what they did. Swapped the snails in different environments, looked at how they changed, and yeah, sure enough, you saw some differences, and that was due to the environment. Sometimes, though, you go out and you sample our population, and there is no difference in phenotype. There is no difference in phenotype, and you can't really separate it. Yet, you're still interested in the amount of variation that exists out in that area. So how do we go about doing that? All right, how do we go about doing that? One way is through sequence analysis. We extract DNA, we sequence it. In some cases, we sequence a single gene. A lot of times, you really want to have several different genes to try to get a better idea of how evolution, how selection, how drift, and all those processes are influencing our genetic similarity and differences. All right, it's great. Prices have really come down. You can sequence large sections of the genome. All right, technology is really advanced. However, the big disadvantage of this is that it doesn't really tell us anything about the adaptive variation that's out there. Yeah, we can see differences. We might identify differences in the code, but is that really adaptive? Is it under selection? Does it confer increases or decreases in fitness. Who knows? All right? But even though we might not know of the adaptive consequences of this variation or these differences, it still informs us about some of the important processes, namely gene flow. All right? So what we can use is sequence our genome and then determine similarity values to calculate something called Wright's F-statistic. All right? And Wright's F-statistic will tell us about the degree of similarity between our populations. So what is Wright's F-statistic? Wright's F-statistic is kind of a play from Fisher's F-value. All right? So Ronald Fisher came up with the statistical test analysis of variance. All right? With that analysis of variance, his test statistic is an F value. All right? There's a distribution, statistical distribution, Fisher's F. All right? And the nice thing about this analysis of variance is that you can take all of the variation in your data and partition it out. So we have overall variation across all of our data, and then we can break it down and say, okay, this is the variation that exists within each of our groups, and this is the amount of variation that exists when we compare our groups. All right? And Wright said, hey, if we can do this with just numbers to perform statistics, perhaps we can do the same thing with genetic values, with genetic code, with genetic similarities, and so forth. So this is kind of what he did. He applied Fisher's idea of analysis of variance to genetic sequences. And that kind of became known as Wright's F-statistic. So Wright's F-statistic is going to partition our total genetic variation into variation that exists within a single population or within the individual populations, and variation that exists between our populations when we compare them. All right? What's important is this equation. So FIT equals FIS plus FST. So this equation here is the mathematical way to say our definition. All right? So we have FIT. That's our total genetic variation. That gets partitioned into FIS, which is the variation within a population, and FST, which is a variation between our population. All right, so I have the figure up on this slide. What we're going to do is just kind of redo it by hand and hopefully just kind of drive home the point. So our populations exist. Our species exist in all these different populations. All right, so our population itself, our species, this is our dashed line. This is our species. And then we have, let's say, population A, population B, and population C. So we go out, we take individuals, we sample individuals from each of our population, we sequence them, we get genetic code. Let's say we sequence 5 genes. All right, we had neutral sites, we had coding regions, non-coding regions, and so forth. All right, and we have our sequence data, and then we start calculating similarity values. How similar are our individuals to each other within a population, and how similar are they to individuals in the other populations? All right, so in the entire population, we have an FIT. This is total genetic variation. Total genetic variation. All right, so that's like genetic variation across our species. And that total variation is composed of all of that variation that exists within a population and all that variation that exists between a population. All right? So with each of these, we have an FST value. It's only a single value. All right, but collectively, we figure out how much variation exists just within a single population. That is our FST value. I'm sorry, that's not FST. It's not FST. FIT and F, what's the other one? IS. FIS. That's inside our population. And then what we do is compare our groups. And that is our FSTs. So it's not just a single FST or FIS value. It's not, we don't have individual ones for the population. We have one value for our entire species. We've got our total, and then that's composed of variation due to variation within populations and variation between populations. So if you've done an ANOVA on Excel and used the data analysis, add-in package, it'll report within and between variation. That's what it reports. And that's what these are. The within is variation within our populations. And the between is variation between. So it's a lot like if each of these has a mean. Let's say it's short, medium, and large. All right? We know how much the short varies. We know how much all of the medium varies. We know how much all of the large varies. All right? We've got ranges. That's your FIS values. And then if we compare short to medium and say this is how much difference there is between short and medium, and how much there is between medium and large, and how much difference there is between short and large, that's the FST. So what you can start to see is that as our FST value starts to get bigger and bigger and bigger, we have more and more variation that's being attributed to differences in our population. That suggests then that our populations, our individual populations, are going down their own evolutionary path and becoming genetically different from all of the other ones. All right? So these FST values are just simply similarity values, genetic similarities. But it informs us of important processes in this population. And for those of you that were watching, this is the exact same graph that's up here. All right, so large FST values tells us that most of our variation across the species is due to differences in the population. What's going to cause that? Number one, isolated populations. Isolated populations. We have these populations, each of them, going down their own evolutionary path. And because we don't have gene flow, they can continue going down their path. They can continue becoming more different than all of the other ones. Different environments with strong selection pressures. If this environment is very different from this, we're going to have a faster divergence because evolution is going to make our populations different. And then small population size, basically due to drift. If we're small population size, drift is going to take over and really start to increase our diversity. So what you should see then is these are very similar to the factors that lead to ecotype formation. And this is one way in which we can measure that. Are we becoming ecotypes? Does it seem sensible that we have ecotypes? Calculate FST values. See how much variation exists between our populations. What happens if we have small FST values? Well, small FST values are like the opposite of the large FST values. Now, we don't have a whole lot of variation between our individual populations. All of the variation is just going to be variation among individuals. All right, so think about heights among the student population. There's a lot of variation between there. All right, so if we did heights here at ASU versus heights at Texas versus heights at A&M, all right, there's not going to be a whole lot of difference in student height in the averages between the different universities. All of the variation is just going to be due to random variation in heights within each of them. All right, we've got short people, we got tall people, and that's the same across the board. All right, so what's going to drive these small FST values? No barriers to gene flow, that's one. Just as soon as evolution starts pushing us down one path and causes us to diverge, gene flow neutralizes it, homogenizes it. All right. Similar environments with similar selection pressures. Now we don't have a separate path. The environment's basically the same. We're evolving along the same path. And then also large population size. So limit the effect of drift. Let selection take over. All right. So large FST values, you've got these three things, these same three things that lead to ecotype formation. If we don't have them, then we can generate small FST values. So we might not know much about the environment. We might not know much about the selection pressures. We might not know much about migration. But if we go out and sample individuals and sequence them and get a large FST value, one or perhaps all of these are at work. Questions? All right. Whoops. One extra slide. Shouldn't have been in there. All right, so we've learned two different ways to look at this variation, this intraspecific variation. All right. Common garden experiments, those reciprocal transfer experiments, those are all aimed at identifying genetic variation and phenotypic plasticity. All right, that's what we try to separate. How much of the variation is due to nature, how much of it is due to nurture. That's our common garden experiment. Sequence analysis, especially today, is becoming more and more popular. All right? And it informs us about some of those processes that lead to ecotype formation. All right? When we sequence it, we can calculate similarity values. And from those similarity values, we can calculate Wright's F-statistic. So if we have a large FST, our populations are genetically different. All right, they're genetically different. In some cases, they may be considered different ecotypes. All right, and then you can go back and we just talked about what could lead to that formation, to that ecotype formation. If we have a small FST, it means that our individual populations are very similar. Right? They're genetically similar. We haven't progressed down that evolutionary path. All right. Questions? All right, this wraps up the material for exam 2. So we are done with this. Review your notes. Start studying. If you have questions, jot down those questions. You can email them ahead of time. I'll take questions over the exam 2 material on Wednesday. All right. And again, if you have a question, it's possible somebody else has the same question. So if you email me ahead of time, we'll go through those questions in class so that everyone can hear them. All right. And if you're worried about being, outed as the person that asked the question, I can make it anonymous. I don't care. All right. So you could email me the questions, or at the start, I'll ask if you all have questions. All right, where do we go from here? We are now moving into populations. And that's basically what we're doing, what we've done in lab, population size. Now we're going to do dispersion this week and next week. All right, and then we're going to be doing demographics. All right, so what we're going to start learning about is descriptors of populations and population growth and so forth. So we're going to move into our population ecology section moving forward. And we'll start that on Wednesday. All right. On Friday, since we have the exam, I'll come here if you plan to take your exam here in the class on your device and your laptop or phone or whatever. If no one's here, I'm just going to head right back to my office. So, but we're not going to have class on Friday because that is our exam day. All right. See you on Wednesday. I guess some of you I'll see later today maybe. All right, have a good one.